Hello, everybody. Good to see so many of you and so many familiar names. And it's officially the starting time, but I'd say we take a couple of minutes, maybe just send a couple of messages in chat. Would love to hear from people, where you are, what's going on. Hope spring or almost summer is treating you well. Hi, Mathieu. Can you hear me well?
Yep.
All right. Excellent. So this is an experiment that's been long time in the making. We've talked about this with Peter Jezzard for quite some time now, that maybe we could do highlights journal clubs, focusing on papers published in MRM. And then this ISMRM in Singapore, we met with Peter, with Mathieu Boudreau, who's on the call, and with Mariya Doneva, who I'm not sure is here yet. And we said, let's give this a try. The idea is we pick a paper, we discuss it, hopefully in an interactive format, and then if this works, maybe we can scale it to include more different ISMRM study groups, different kinds of MRM papers, and hopefully build a community around it. So I'm just buying time because I see that people are joining. We're already at 46, 47. I'm really happy with these numbers. And hopefully in a couple of minutes, we can get a discussion going. The paper that we chose today is the T1 challenge paper that's been a long time coming, and we'll hear a little bit about its genesis. And also what I'm really glad to see is many of the co-authors of the paper joining here, because we would like to use this opportunity to discuss with them and see everybody's opinion on the results and the way that we presented it. As I said, Mathieu is here. So Mathieu, good to see you. Peter is here also. So Peter, thanks for joining and for making this happen. Where are you at the moment, Peter? Are you at work?
I am. I'm pretending to be in Northumberland with a castle behind me, but I'm really in my office, yeah.
All right. Still great background. And thanks for advertising this in the MRM subscriber letter. And thanks for joining. I'm checking out the chat. People still haven't started posting things, but maybe you could just, give us a hi. Feel free to turn on a camera if you want to speak also. I'll just call on a couple of people that turned on their camera. I assume they're comfortable talking to us. Yves, good to see you.
Morning, Nikola.
You're having your morning coffee, I assume, 8:30 Montreal time?
Yeah, it's still morning here.
Cool.
Nice to see everybody. Thanks for coming.
Yeah, thank you. Maybe you can say a couple of words about the award you received for the 10-year-old paper that we co-authored together, because it's kind of a prequel to what we're discussing today.
Yeah, it's kind of an interesting circle, isn't it? So last week I was in Regina, Saskatchewan, which some of you may have heard of. It's a sort of mid-sized city in the middle of the prairies in Canada. And I was attending the annual meeting of the Canadian Organization of Medical Physicists, of which I am a member. And we received a very nice surprise in the weeks leading up to the meeting that our article from 2015, I guess we submitted it in 2014, it got published eventually in 2015, which was kind of the precursor to some of this work on comparing T1 mapping methods. And this was work that Nikola spearheaded, and Mathieu and I were involved. Mathieu, I think, was a PhD student at the time, and Christine Tardif was on that paper as well, and so was Joelle Barral, who had published the original, who had led the publication of the original method for short TR, sort of well-modeled inversion recovery T1 mapping. And so we went ahead and did a study and comparing T1 mapping methods on one scanner using phantoms and in vivo participants, and it got published in Magnetic Resonance in Medicine. The findings were obviously interesting enough because even today it's still gathering citations, this paper.
It's about T1 estimation across different scanners. So it's talking about.
Sorry, I'm just going to mute.
Oh, Okay, I thought maybe there
I
was a
think
question.
no,
I wasn't sure. I was going to
somebody
listen
just
to the end. Okay.
unmuted.
Okay, thanks. Right, so we did a comparison, got published in Magnetic Resonance in Medicine, and talked about playing the long game because 10 years on from that publication now, we received a prize for it, which is the article published by a comp member and performed in, work performed in Canada. So it's pretty specific, but nonetheless, the paper has gathered almost 400 citations by now. Well, it's well above 300 anyway. I don't know if it's close to 400, but it'll get there. And we received the Michael S. Patterson Publication Impact Prize from the Canadian organization. So it's quite a nice surprise to come around. I'm sorry if there's a bit of background noise. There's some construction going on in the neighborhood here. But yeah, it was a nice surprise. I was on site for the meeting anyway, but I was happy to be there to accept the prize on behalf of the team that published the work. I guess I didn't mention who the senior author on the work was. I mentioned every other author, but the senior author who enabled the study and made sure we had scanner access and made sure we all had funding and postdocs and PhDs and whatnot was Bruce Pike. I don't know if Bruce is joining us today. It's a bit early in Calgary, maybe. He's not in Calgary.
It's too early for him.
But Bruce, generously supported this work and supported it, and we managed to get it out there. So it's nice to see. Now this paper, which actually the title borrows from the original one, right?
It does, yeah, exactly.
Still seeking common ground on T1 mapping, and so it's nice to see this come around now with a multi-center study that has been published. Right.
Yeah. So the threads are there, and actually the first thread is the original Joelle Barral article. So Joelle is with us today, and I'm really happy to see her because that paper has really inspired a lot of follow-up work, and hopefully we can hear from Joelle her reaction to the current challenge, but also just a bigger discussion about where we're going with T1 mapping. So Joelle, good to see you. How are you doing?
Yeah, thanks, Nikola. Hi, Eve. Hi, everyone. Joining from London today. It's really a trip down memory lane for me, right? Like I'm now with Google DeepMind. I've been in this whirlwind of Gen AI for the last 2 years. I jumped off my MRI career and expertise maybe 10 years ago when I joined Google, and I was then focused initially on surgical robotics and now closer to the research again. So it's really wonderful when things you've done years and years ago come back to hunt you in a very positive way. And I think this is the case, right? I wrote a paper when I was finishing up grad school entitled "A Robust Methodology for In Vivo T1 Mapping." And I remember Nikola and I really started that work because we were frustrated that an MRI, like a magnet, wasn't a scientific instrument, and that we were reading a bunch of papers, and the T1s were always big ranges, and it was really puzzling that we had those big ranges for T1. Even in healthy subjects. And so we started looking at the methodology and kind of pulling that thread from how do we measure it, and is it is there a science in this? And so that paper was really trying to get to a reproducible protocol that people could then use, right? And I thought it was actually you would think, right, 2010 MR is already fairly mature by that time. But actually not really. And what was really surprising to me at the time was that a lot of things we took for granted were not that precise or not that well known technically. And I was hearing about the news and this award. I was really reflecting on the time we are right now with generative AI. It's not a science. There are many things we don't understand. It's not a precise instrument either. And I can only hope that some of the researchers, and I'm surrounded here by thousands of them, really ask those scientific questions, right? And take a very scientific approach to trying to figure out what our models are doing, under which circumstances, against specific benchmarks, when they can leverage specific data, and really, again, pull that scientific thread little by little. I trust, we'll be in a very different place 10, 20 years from now. But I'm curious, like which of all the papers that we collectively publish will, kind of ask the right question to be still meaningful many years down the road. Anyhow, it's really a pleasure to be with everyone, and look forward to the discussion.
Yeah, thank you, Joelle. And really, a lot of this work has been directly inspired by you, and I'm glad you took the time. So I think this sets the stage. Lovely to see many of the other co-authors. Feel free to, speak up, say hi, and would be great to hear from you either now or maybe after Matthew's presentation. I'll just kick things off by just talking to Matthew a little bit. Again, this is something that we have discussed quite a bit among each other, but it's nice to give the context for why this challenge happened. And I will just, say what we finally remembered, which is that this was the second challenge of the Reproducible Research Study Group. The first one was organized by Florian Noll when he was running the study group, and it was on Klaas Prusman's sense work. And then this was the second challenge, and we said, let's do something about quantitative MRI. And then I brought this up with Mathieu, and this is before the pandemic, so it's quite a long time ago. So maybe Matthew can give us a little bit of context and introduction to how this whole challenge came about.
Yeah. Well, I was invited to, I guess, prepare or propose part of the challenge. I think the main idea came from you. One question I had for you actually, Nikola, was I guess how did the quantitative MR study group joined along? Because the first I think challenge was just the reproducibility study group. Was it?
Yeah.
Do you recall?
So we wanted to find a paper that first was a influential paper, and second was a little bit different from the sense work in the sense of we wanted to bring more quantification in the mix. And I've had this desire to unpack quantitative MRI, and I've been focusing on T1 mapping quite a lot. And then I thought maybe we pick a paper where I know the first author, I've been involved with it, and whatever comes out of it, we will understand that this is in good faith, that we're actually trying to make science more reproducible. And the other thing is that the original Barral paper is one of these rare papers that actually shared code in 2010 together with data. And it's really easy to take that and apply it to different kinds of data sets. So to me, it was, let's pick a paper that I'm familiar with. Let's pick a paper that, regardless of what the results are, the co-authors won't mind. And then let's run with it and see what happens. So that was my reasoning. And, I know Joelle was involved in the original discussion. Maybe Katie Keenan, if she's here, they could give a little more context. But that's what I remember from 5 years ago.
Yeah. Well, I think before I set up the challenge, we had some meetings with Katie Keenan, and I remember we had some meetings with Paul Toss, I think, because he really wanted to try
we
to.
did. Yeah.
I think one of your main focuses was to try to hit on his conditions for what was it called exactly? Like
Yes,
like
the gold. We wanted to give medals
the
for
gold medals.
reproducibility.
Yeah.
Yeah, exactly. Right.
Yeah. And so that was the original idea, and we had written up for the NIST Phantom, or the NIST CaliberMRI Phantom. That was just produced, and yeah, I had just written up a proposal, and then in the middle of it, we had kind of said, well, maybe not everybody has access to that Phantom, so if people wanted to, acquire human data, we opened that up as well, and the focus was on reproducibility and openness. So we said, from the beginning, we're going to make all the data open, code open, so make sure that you get all the permissions to do that, and then we launched it. I think the idea was that people were going to scan in February and March of 2020, and that got cut short. But yeah, from that point on, I'll talk about a little bit more the specifics of the paper afterwards. But I think that was the main timeline, and then we were on a steamroll up until ISMRM that year to present the results from what people had time to acquire prior to the lockdowns.
This is right before the pandemic. I just shared the call. This is like December 2019, and we tell people, acquire data until March 2020.
Mm-hmm.
And of course, life interfered. So this has been a long journey, almost 5 years now. But that's the original call, and we paid a lot of attention in the wording of that call because the question itself had to be unambiguous. So I think, Matthew, you will mention what the question was, but we said other studies have always done things in a very controlled environment. For this study, we're just giving you a paper that was published. And I think, the results reflect that. So maybe we can start with a brief presentation from Matthew. I think it's going to be around 10 minutes.
Yeah.
And that will leave us plenty of time to, go back and discuss between each other. So Matthew, when you're ready, go ahead.
Sure. Share my screen there. Sorry, I'm juggling tabs. There you go. You see my screen?
Almost. Yes, now we do.
Okay, great. Yes, I just took a screenshot of the paper that was published. I guess it's just been a couple of months now. And yeah, we had a lot of collaborators. I think 41 authors, and many of them got help from a lot of other people. So really, you know, great appreciation to everybody worked on this and took the time to prepare the data and the acquisitions. So in terms of the paper, the question that we specifically focused on was touching on what you'd mentioned, Nikola, was specifically like, are the acquisition details that are provided just in a paper, so like in the written document, so by Barral, are they sufficient to ensure reproducibility across independent research groups? So I mean, the idea that when I wrote the challenge was to simulate what we do in research, all the time. Somebody publishes a paper, we, take a look at the methods and then try to reproduce it. So there were some other papers that had done it in a more controlled environment. So I know Katie Keenan did one, and I forget the authors of the other group, but there's a few groups that did it where they had more control. So really like a shared protocol that would share across sites. But we really wanted to see like how people were going to handle like working on this independently. And in the paper, we did this by comparing the intersubmission variability with an intrasubmission variability in phantom and brain. So we got lucky, and there was one group that acquired in phantoms, and a different group that acquired in brains that acquired very, very large data sets. So they had enough time. So in the phantoms, it was a group in Germany that acquired at 7 different sites using their shared protocol. And then in the brain, I think it was in Mexico, where they acquired on 20 or so subjects on 2 different scanners. And so we were able to calculate the intersubmission variabilities for these 2. And then across the other, I think 18 or 19 submissions, the intersubmission variability of T1 in different regions. And just to cut to the chase, we found that in our case, the intersubmission variability was twice the intrasubmission variability in both phantom and brains. The measurements were fairly, overall accurate. There were some outliers, but we could definitely see that there was more variability when acquisitions were done independently. And so our conclusion was, if you communicate just via paper, the acquisition details, the pulse sequence details, it wasn't sufficient for different groups to reproduce the measurements precisely. This is Joelle's paper. As it was mentioned, it was published in 2010. A few features of this paper was that, she had used, I think, the Kramer-Rao lower bound to optimize the parameters. And also, this model, unlike other inversion recovery T1 mapping models, doesn't assume that there's full recovery at the end of each TR. So it's not there's no long TR assumption. You don't need to have TR 5 times T1. That takes it into account in the equations and the fitting. And should share the code that we had used in this challenge specifically. This was the protocol details that we had shared with the authors. So we'd said, here's what we want you to acquire. This is the amount of details that we're going to give you. And if all the other details, try to do as you would in a real, in a real study. If you want to use fat saturation, whatever, make those decisions, but try to stick with this. Some groups were not able to get these values exactly on some scanners, and so we said, get it as close as you can on your scanners, and report what you've done. And so we had all those with the submitted data, exactly what they had, the exact values that they had used. So we had 18 submissions, so 14 acquired phantom data, and then a separate, or not separate, but 10 acquired human data. There was some overlap. Some groups had acquired both. The phantom is the ISMRM NIST phantom that's produced by CaliberMRI. And so there was 2 different versions. For the analysis, we had stuck with one of the versions because the T1 values can vary for the different spheres on the T1 plate. So here's the site that had acquired on 7 different scanners. So we did an intra-submission coefficient of variations for the different ROIs of the different T1 values of the sphere here, and then we did the inter submission variability between the green sites. And then same thing with human. The Mexico group had acquired on a lot of different subjects on 2 different scanners. We did the intra submission variability in the orange, and then inter with the remaining. And so for all the figures, the color coded is the same. So the orange is going to indicate what the sites that were used for the intra submission variabilities were. So this was, I think, I'd say the main figure in our paper, which was some violin plots of the mean ROI values for the sites that we had used for the inter-submission variability, so in green, and then the distribution of values that was in the intra-submission. So it's, I mean, quite, I think, clear here that there are some outliers in some cases, but they're overall, when I showed in the scatter plots afterwards, the intra-submission was quite more precise in the repeated measurements, or reproducible across the sites that they had done. And same thing in the forebrain regions that we had looked at. So this is somewhat the same data, but just displayed differently. So these were scatter plots. On the A on the left, this was a linear plot with the temperature corrected reference T1 values of the phantom. And then here's the log plot. This kind of takes away some of the variability that would be biased just because it's a linear. From the high values, we could see that's quite accurate, especially for the intra-submission. And then the errors in terms of percentage is on the right here for the phantom. For the human, here we have the GNU on top, splenium, cortical gray matter, and deep gray matter. And the orange site was the multi-subject on 2 different sites. And so here as well, we can see there was less variability. We'd shared all the data. So the data that was acquired, the T1 maps that were fitted, and the ROIs that were used are all on OSF, and all the T1 fitted pipelines, the analysis code, even some of the manuscript preprints are available on our GitHub repository. And we had also used this for the submission process as well. So all that is tracked, and there's a history of that. As well, we had submitted this before submitting to MRM. We had submitted this as a preprint to NeuroLibre, which provides somewhat of an interactive version of the manuscript. So all the figures can be interacted with. Aga Karakuzu, who I don't think is able to join today, who's my co-first author, developed this dashboard. I'll show this on another slide as well afterwards. So you can just go on this website. You can look at the DOI and access that website, and go in and look at the figures. You can look at the code that was used to generate the figures that will be above the figures as well. And so this provides some, I guess, enhanced information that you could get, you might not be able to get just from the printed paper. In addition, here, this provides us access to a binder environment. So a binder is a way to run a virtual computer in the cloud and access it through a web browser. And here you can open the notebook that was used to generate this interactive preprint, and you can go in and look at the raw values, look at reproduce the figures. You can run them all again, run the analyses, and this is all running the data on some servers at McGill. So this is just an example. If you've never seen a Jupyter notebook, this is somewhat of the workflow, or what it looks like before we built the NeuroLibre preprint, the HTML. It'll just scroll down until we see one of the figures that was reproduced. There we go. And lastly, the last thing I wanted to mention was the dashboard. So this is a website that anybody can access. And here again, you can take a look at the values, some statistics from all the sites that were measured. You can look at different summaries if you want to just look at some vendors. So we wanted to, make the data more accessible to anybody that wanted to access it. You don't need to be a coder or anything like that. You can just go in here and directly look at the values, and it gives us both for the phantom and the human. So here are the ROI values. So that's about the summary that I wanted to give to bring people up to date if they hadn't read the paper, and hopefully we can have a good discussion now.
Great. Thank you, Mathieu. So I would like to ask the co-authors of the paper, those that are on the call, if they could, let us know, maybe turn on the camera or just, give us a wave. I just want to get a feel for how many of you are here. Perfect. Thank you. And I think that the best way to do this, because there were quite a lot of us, it was a challenge to get behind a common message. And, the way that this article evolved was really with many iterations where first it was just a big data dump, and then it filtered, distilled into a more coherent message. I would like to hear from the co-authors what they take out of this paper before we open it up to the rest of the audience. So anybody that contributed, please let us know if Mathieu missed something, if you want to add your own opinion, and maybe comment on how we got to this final version of the manuscript.
I think Yves had a student that had done a little bit more work with that data, right? Do you want to lead this off, Yves, maybe?
Oh, it's been a while. So yeah, a few of my students were involved at the time. I looked at this challenge. In fact, maybe this is not that scientifically interesting, but I did look at this challenge as a way of getting a few students involved in some international collaboration work and to learn about T1 mapping. It's still relevant to my lab in another context. We do T1 mapping for other applications now than brain. But yeah, it got them excited to collect some data somewhat on their own. They were just sort of training to use a couple of MR systems. We actually were one of the sites who submitted data from 2 different MRI scanners, and we submitted phantom and in vivo data. And indeed, one of my students who was involved in the challenge, now I remember, she's a medical physicist here locally now, but she included our data, the contribution to the challenge, in her master's thesis as sort of a demonstration of cross-scanner reproducibility. So she played around with that a little bit. I have to say that just with that subset of the data, I don't really remember the results of the analysis. She finished her thesis in 2020. So as you know, this came sort of right at the right time. She acquired the data, and then the rest of her thesis actually used some of the methods. And as part of her thesis, we also compared inversion recovery and saturation recovery techniques on one scanner. In fact, we were driving towards helping out some colleagues who were doing T1 mapping of nanoparticles. They were looking at T1 relaxation effects, relaxivity effects of nanoparticles as a potential contrast agent. And the challenge served as a way for us to benchmark our T1 mapping, to know we had good confidence in our approaches for T1 mapping. We did play around with the data a bit. We I think along with Guillaume Gilbert from Philips, who's also based here in Montreal, we noticed sort of one of the slight, it was a very minor issue, but it had a bit of an impact on the parameter selection. The sort of native inversion recovery sequence of the Philips scanner wasn't able, I think, to reach all the way down to the required TR given the selection of TI. So there was a little bit of fiddling around that. We ended up using the slightly longer TR. But yeah, that was some of our work. The result, the details kind of escaped me a little bit. I could have gone back over Anastasia's thesis. She's a co-author on the paper, and the thesis is actually available through the McGill eScholarship website. But yeah, we did a little bit of sort of parallel analysis, complementary analysis. We looked at, as I said, saturation recovery, inversion recovery, and VFA, I think, in the same phantoms. Now that's a limited data set. It's just on one scanner. But yeah, that was our contribution to the work, and noticing some of those technicalities, if you want, along with Guillaume Gilbert, who was a big help in getting us started here.
Oh, Nikola, you're muted.
I am. Yeah, thank you, Yves. So I see that Mariya unmuted, and I'm curious to hear, because I know that she was involved in this kind of intra scanner variability. So Mariya, please.
Hi guys. So yeah, we did that. I think we were the 7 different scanners in Germany, right? And I think I don't know. I think I was much more positive about the messaging than you guys, like, in terms of, hey, putting in the title, paper is not enough. I think it's amazing what we have achieved in terms of reproducibility, considering that, as Yves said, it wasn't easy to get exactly the same parameters. We also struggled with that. We also, the first scan that we did at my site, it was like the values were way off, and we was like wondering what was going on, and we figured out it was freezing. It was just really the temperature was way off that day. And we scanned again, like, with room temperature, and like we were there. So it's like, okay, you have all these variations that we are not accounting for. We cannot avoid some of variations in the sequences, and still, I think what we achieved was amazing. So I think, there is hope. I think that's a good messaging. So that's from my side.
Yeah, I agree. And this is where, maybe we were a little pushed by reviewers in this direction. But, we wanted to make sure that we give this paper fair treatment. In a way, the results are good. It's actually, as good as you could expect, given that, Philips and Siemens and GE don't even operate on the same main field strength, like they're not all at 3 Tesla. Not to mention all of the different kinds of variabilities that, come in with spoiling, with gradient performance, and so on. So I think it's as good as it can get. And I feel that the discussion does this justice, but I'm glad that you argued that angle because in a way, maybe 10% variability is actually good. The only thing we showed is that because you coordinated, your results were closer. And then if you didn't, then, you would expect bigger variability. But that's an absolutely valid point that these are actually pretty reproducible findings, except when you coordinate, obviously you share something that's je ne sais quoi, that's a little extra from what is actually in the paper.
Yeah, well, we coordinate because we had one phantom, so you had to, travel through Germany with this one phantom, right? So it's kind of like if you have set it up, you don't do it differently, but I don't recall if we had to adjust anything in terms of parameters on different systems. So these were different systems, so you could expect that maybe the software is not exactly the same, so maybe there were some variations in the parameters as well.
Yeah. Now, this was all Philips for the Phantom.
Yes.
But then in the human study, actually the intra data set was Philips and GE data, if I'm correct. Mathieu?
Yep.
So there, it was actually 2 different scanner versions, and yet, it showed that, the coordination is something that does help reproducibility. One thing that we didn't touch on is what are these factors that contribute to bigger variability? And this is something that's in the discussion, but maybe some of the co-authors can discuss because that was an internal discussion that we had while we were writing it.
Yeah, it's a shame Aga's not here. Maybe you want to mention Aga's because Aga shares some features with Aga's paper where he did multi-vendor with the base pulse sequences and then with the vendor neutral pulse sequences. Do you want to mention that, Nikola?
Yeah, so I think that's one of the things that's missing from a lot of the quantitative MRI work is that the details of the pulse sequence are described in the PDF, but the actual code is not shared. And a vendor neutral approach, especially something that takes into account the pulse-seq standard, tries to make it easy to deploy the same sequence on different scanners, is probably the right way to go towards treating the MRI scanner as a measurement device. And I know that quite a few people here are playing with these different vendor neutral implementations. And as a matter of fact, there will be a follow-up work for this, which is the cardiac MR study group decided to do a T1 mapping challenge by sharing a pulse-seq description. So, in 5 years' time, we're going to get together again and we're going to say, and now we did a challenge with a shared pulse sequence implementation. Let's see whether that makes things any better. And I don't know if any of the cardiac MR folks are here. Adrian Campbell-Washburn suggested this, but it's an exciting follow-up work that I think would show. He says hopefully fewer than 5 years. I'm going by historical precedents, but I think we're getting to a point where we realize all of the things that matter to make sure that the measurement is more precise. Vendor neutral implementation will go a long way. Any other comments from the co-authors? I see that Mathieu is here, Risto is here, and if not, I'm happy to open it up for discussion with the larger community. Yes, Risto, please. You're on mute. Yeah.
Yes, good afternoon from the Central European time zone. Yeah, this is very interesting, but on the other hand, nothing unexpected, in a sense that you have chosen possibly the most complicated biological measure by MRI you can measure, and that creates many issues. And my simple question is that, is this what you have observed, something specific to T1, or how about any other measures that are quantified by MRI? It just reminds me about the Dementia MRI conference, probably about 10 years ago, about volumetry. And in an EU consortium, the conclusion was that not even the volume of a hippocampus could be reproducibly measured across the sites.
Thank you. Very provocative and very timely. I would be curious if other people have a reply, and otherwise I'd be happy to jump in there.
This isn't quite a reply, but I suppose something somewhat related. I wonder how much of that variation in the hippocampal segmentation was because of segmentation differences, and that wasn't something that we looked at in this work. I think did Mathieu do all of the segmentations? I think it was one person that did all of them. Whoever had that task, I'm really sorry.
Yeah, no, it was me. I didn't segment it. I just took ROIs in the main regions. One of the challenges that we had, because inversion recovery takes a long time, everybody acquired, or almost everybody acquired, just a single slice, and not everybody had the same slice positioning. And so that was one of the challenges, and probably one thing that really caused some variability for the human side of the data. For the Phantom, that was somewhat automatically segmented. Gineko and Haddad had written a registration script, and we had some mask information from NIST. So that one was a little bit more controlled, I'd say, than the human one. But yeah, definitely part of the challenge.
Do you think that the region of interest segmentation that you did, because it was reliant on the positioning, which was different for different sites, do you think that is a sort of reasonable representation of how segmentation methods might vary across sites anyway? Or do you think because you were doing all of them?
No, I wouldn't infer anything on segmentation tools and segmentation. I think they would have been more robust. If we would have had 3D volumes, I would have used them. I wouldn't have done it manually for sure. And I think it would have probably reduced some of the variation that we saw. So we mentioned that in the paper as well. Yeah. Thanks.
And to answer Risto's comments, you're right. T1 is complicated, and the problem is that our initial reaction is, if we're talking about quantitative MRI, people say T1, T2, because they are the two relaxation times that we learn in a basic MRI course. And it's only after having been working in this field for 20 plus years that I realized, yeah, T1 might not be the best candidate for this. The problem is that many other quantitative MRI routines rely on T1. I've been doing quantitative magnetization transfer. What do? First, you measure T1, and then you apply many MT pulses, and you fit it into a model that assumes that T1 is measured well. So then it becomes turtles on top of other turtles that, show that if the variability at the lower level, the lower turtles, is big, you will only amplify that noise in subsequent models. Now, there was a session in the ISMRM in Singapore organized by a couple of people from NIST and EPL in the UK, and they invited Els Veermans to talk about diffusion. And we think of diffusion as this kind of complex, multi-variable fitting, but there are certain simple diffusion measures that may be more reproducible than T1. For example, ADC, something that is even clinically useful. So maybe that's an interesting point to hear from other people. Are we chasing the wrong quantitative MRI parameter? Are there other low-hanging fruits that may be a little bit easier for people to reproduce?
I don't think you are chasing the wrong, but you are chasing the most difficult, or one of the most difficult.
Yeah.
One thing, if I could mention, at ISMRM, Jacob Aslander has had a poster. I wasn't able to make it because I had to leave unexpectedly, but where he had reached out to us about the 2015 paper and asked us for the very specific pulse sequence diagrams, if we still had access to them. And what he did was looked at the MT effects on a lot of different published T1 mapping sequences and works. And I think he found that something like 80% of the variability was due to the fact that we weren't taking MT effects into account from the pulse sequences and how the MT effects varied between the pulse sequences. So yeah, T1 is difficult because of a wide range of other interactive phenomenons that can affect relaxation. But
yeah, Yves?
Yeah, you make a good point, Mathieu, about magnetization transfer, because depending on how you code the inversion pulse for the inversion recovery sequence and the selection of the initial TI, the shortest TI, there can be quite a bit of difference in white matter, especially because that's where magnetization transfer effects are greater in brain, but it's also present in gray matter. And you don't see this in pure liquid phantoms, but the selection of that 50 millisecond TI can make a big difference. And if you just shift it over to, say, I don't remember the exact, I was looking at this kind of around the same time as this work was, as the original, sorry, the original Common Ground work was going on in sort of 2014, 2013. And we noticed a big difference between sort of mapping T1 with 50 milliseconds or 150 milliseconds for the first TI. If you shuffle the other TIs, it makes less difference. But so I published, I think I was a co-author on a paper with James Ryu, that looked at this and sort of playing around with the first TI. And if you look at the MT work of the Nashville group, the Vanderbilt group, right? This is Dan Gotchberg. For years, they've been studying the magnetization transfer effect using these specially selective inversion pulses. So they try and invert just the water resonance and leave the restricted resonance alone. And they, in fact, exploit that short period between sort of 0 and 100 milliseconds to get, in fact, all of their MT effect from that set of data. So it is a factor in there. And if you don't pay attention to the tissue you're measuring, and then this particularity about the method for inversion recovery, you get variations, and then the style of inversion pulse. So these are probably things to look at. I mean, I don't want to say look at again, but maybe to integrate, right, the data we've collected, and then the observations from Gotchberg in magnetization transfer when it comes to maybe standardizing these. And I think Hristo makes an interesting point at possibly going after a difficult parameter, but I also think he's very right in saying, I don't think there's a wrong or right parameter. Really, this is about figuring out if T1 has value for in vivo work, which I think has been demonstrated by a number of studies in various applications. But it's more about, checking out how we can better standardize the measurements.
Thank you, Yves. So Olivier raised the hand, and Mathieu afterwards.
Yeah, thank you. I think this is a very important point because, as you said, I mean, MT effect explain a lot of the T1 variability. This is actually what explains why measured T1 is lower in inversion recovery-based sequence as compared to variable flip angle. But there is also an interesting thing is that when you do QMT measurement, you need an independent T1 measurement. And now we get to understand that when you do T1, you would need also to account for MT. So I really believe this should drive us to measure both simultaneously, which actually we did here in Marseille. Other people have done it, and we are working on improving the biophysical model to really improve the stability of that. We are close to getting similar value with MP2H versus VFA, which I think it's a nice achievement that we have at least removed the let's say the first order contaminating bias somehow. And I think this is important that we should, it's not that neglecting T1 or focusing more on diffusion, but we should really probably think of the different parameters as estimating them jointly, because we know we have, I mean, ideally we should get orthogonal information from different modalities. And we would like to infer tissue biophysical properties with this added information. But if we keep estimating each single modality differently, we're not doing the let's say the math, the statistical well. We should really, I think, combine all the model together, which requires some work first to kind of well, establish the let's say the minimal requirement model, which has just enough complexity to account for the data variability, but not much, so that we would not increase the variance of the estimate. So I really think this is a very important topic. And yeah, I don't know. I mean, I think it gets more and more popular nowadays that MT and T1 are basically different metrics of same mechanism. So I don't know, why not? Because they both focus on longitudinal magnetization, basically. So why not making a joint T1-MT contest? That would be interesting to compare different models and implementation.
If that's a offer to volunteer, then by all means, that would be a great idea. So thank you. Thank you, Olivier.
Well, I would be happy to help. I've not been involved in the first one, but if I have guidance, I'm happy to put energy into this. Yeah.
I think the one thing that is I believe very important here is that the data is now available. So people can pick a small subset and try to explore, because these are questions that I think the data set may be able to answer.
Yes, except that, well, the easiest way to combine joint T1 and MT is to get at the same time T1 sensitive data and MT sensitive data. So basically, the easiest thing to do is to combine VFA with Z spectrum or inversion recovery with Z spectrum. So, well.
I agree.
Yeah.
Mathieu, Greg Sellers.
Yeah, so I wanted to just pick on one other thing, which actually is the gold standard and what we assume to be the gold standard in terms of the T1 and like value in the phantoms. And I'm just picking on this in particular because Mariya was saying that they didn't really standardize across the different sites when they did their 7 scanners, but actually it is the same phantom. And what we found when we were doing an analysis in parallel on MR fingerprinting and also variable flip angle, and then this was an additional acquisition for us, and we realized that especially 1 or 2 of the spheres were off from what the manufacturer had said that they should be, which led to a whole new discussion with the manufacturer and them updating their values. But I wonder how much of that better accuracy, so to speak, from the intra whatever kind of variation is due to it being in the same phantom rather than it being because it's the same people doing it, kind of thing.
One thing
Mathieu,
I could just briefly mention is that there were other groups that shared phantoms as well. So for example, we had shared the data I think that Aga had acquired was the same phantom that the data that Yves had acquired, and there was another group in Montreal. So in Montreal, we had shared the phantom across different submissions as well, and there was variability, like higher variability there than what had seen in Philips. So I don't think it could explain all of it. It definitely helps for the Philips side, but yeah. That's just what I want to mention.
But
Nikola.
this is related to Tom Griesler's question in the chat, which is, what about drift in values? And for example, we, as in Montreal, are the site that got the first new version of the NIST system. So the values are actually different. If you take a look at NIST values between 1 and 41 for the phantoms, and phantom number 42 and on, there is actually a discontinuity there. So even that is something that, we need to be aware of. And then drift, these values are probably going to change over time. I know that CaliberMRI is trying to characterize that. So by all means, the gold standard itself and the gold standard phantom itself is a moving target. That being said, I really do believe that this article is the best we can get to a real gold standard measurement. And in phantoms, it's much easier to be, close. In humans, we observe the same thing. We had a site that coordinated across different vendors, and we saw that, the values were closer together. Now, human variability is even bigger. So there, we're not even comparing apples to apples. We're maybe not even comparing fruits. But we do observe this trend of if you put, sites to work with each other, it's easier to get similar values. And I really hope that some people will do some follow-up work, using the data that we shared, using the code that we shared to try to tease out some of these second order effects that are probably driving a lot of the variability. The phantom number and looking at drift by the age of the phantom is something that would be really easy to do with the available data. If anybody wants to do sub-analysis, that would make me the happiest about this challenge. Cool. So, yeah, Richard, go ahead.
In phantoms, use spectroscopic methods to see the aging rather than imaging.
Yeah. And I know that, the validation is originally done in an NMR system, but I think it would be interesting to do spectroscopy as follow-up work. So this leaves us with about 10 minutes of the regular time, which I think is perfect timing because this is an experiment. We did it not knowing how it's going to work out. And given that I've been very involved in moderating and organizing all that, I would like to hear feedback from people. Especially, I would like to hear from Peter, whether this is a format that could carry over to other papers in the Highlights MRM Journal Club format, but also from you that attended, what we could be doing differently, or whether you have certain ideas for papers that we may want to discuss in this format. So Peter, maybe you first, and then other people on the call.
Yeah, sure. I mean, I think it's been a fascinating discussion. I think that it's very valuable. Some of you may remember that we did this experiment some years ago. I think Mark Griswold used to host a journal club for MRM some years ago. And I thought it was very good. And really the reason that it didn't continue was because of Mark's time. And so Nikola and I were talking about how this might sustain in the future. And one idea that we had, and we've again be very grateful for feedback, was that we could invite study groups in turn to pick a paper that they think is worthy of discussion in a forum. They could form the panel that discusses it. We'd invite the authors to briefly present it in the way that Mathieu did today, and then open it up for questions. And I think that would be very valuable to do, once every month or two. Doesn't need to be very, very regular, but I think it'd be nice, and it might draw a different audience each time, and that would be great too. But that's our thoughts, but we would very much welcome some feedback.
Thank you, Peter. Other comments about the format of this journal club, and then, maybe we can read some of the comments in the chat from Stefan and Callie. Every time I do this on Zoom, I have PTSD from the pandemic. So I wasn't even sure if people are going to show up because it just reminds me of, the period when we had to do this on Zoom. But maybe it's, forgotten and, we're okay with doing something online like this. How did people feel about the duration? Should we have longer presentations? Should we try to motivate a discussion early on with something provocative, or is this the right way to do it, just kind of in real time, come up with discussion topics?
Well, one question I had, and I guess this wasn't preparing, was if there's a preference in terms of having this kind of open Zoom call or more like a webinar format where there would be like the panel of, co-authors. I think I like this call. I like that anybody could join in the discussion, but that would be my question.
Yeah, we decided to do it very open. We felt like, this was not publicly announced. The link wasn't. So we knew who was registering, and everybody's been a very constructive participant. So I'd like to keep it this way until we have a problem. Because, the moment you have a problem, that actually, makes you realize that you were preparing for something altogether different. I would be okay with keeping it open and allowing everybody to speak, but, please let me know if anybody disagrees. Maybe this is a moment where we can just bring up the comment from Callie. So she's from CaliberMRI asking, if anybody has problems on the values or wants to help, please reach out because it is their phantom that we were using. And then Stefan with a very provocative, but I think accurate comment on T1. Can MRI be a measurement device? Because at the moment it's not. I'm not losing hope. I feel that there is a way to constrain this further and further. But as you see in the chat, people do feel strongly that this may not even be feasible. Anybody want to comment on that? I would put Stefan on the spot, but he doesn't have the camera on. So All right, we'll leave it to a chat discussion then. But I think we're all very aware. The problem is that AI algorithms are not. They're treating MRI scanners as data. They're treating MRI data as something that they can just put in a model and come up with results. So quantitative MRI is seeing this renaissance, but I'm really worried that we may be measuring things that are not measurable, which is what Stefan's comment implied. All right, so we have 5 more minutes, but I think this may be a good stopping point. Thank you, everybody who joined. Stefan, you turned on your camera. I didn't want to put you on the spot, but I would love to hear more in the remaining few minutes.
Yes, so I'm an engineer by background, but I spent past 30 years working in hospitals, with exception of 1 year where I worked at the technical university, and medicine is just intrinsically imprecise technique. Even things like body temperature, we can argue that thermometer is an accurate measurement, but depending where you stick it in the body, you will get different measurements. So it's not really the device, it's the standardization of measurements and being able to track protocols and compare the same to the same, which are really meaningful. And my comment is just relates to effectively the fact that if you put thermometer into the ear or under your arm or in your mouth or in other parts of the body, you will get different temperatures by 2 centigrade, which is a difference between norm and pathology for a single measurement. The same is with T1. I started working with T1 mapping in 2008, and I was relatively then a newbie to MRI. I came to MRI hoping that it will resolve and feed all the parameters I needed for brain modeling, and then it all turned out to be imprecise, inaccurate, and over-promising. The blood volumes are not blood volumes, blood velocity was not blood velocity, but realistically I realized it's not because of the measurement, it's just because there is no such a thing as a blood velocity in the brain. It depends which artery you look at, doesn't it? Okay, in large artery it's large and then it progressively decreases. And of course, MRI doesn't have ability to measure precisely one specific point. There's always some sort of average of very specific media. For T1 mapping, again, because it's so multi-compartmental and multi-level, depending how you set TIs, you will get different T1. Because you will be looking at it more precisely or be biased toward the short T1 species or will be biased toward the long T1 species. So intrinsically, it will depend. So for the past few years, I'm trying to sort of promote standardization, open methods. I really appreciate OpenMOLI, although I'm somewhat scared that it takes us methodology-wise back to 2003 about. So I mean, there are trade-offs, and I look with great interest what's happening in the field. We try to sort of promote stability, which is extremely hard. I try to stop. I mean, we work mostly on Siemens, and I try to sort of follow up what happens on the platform. It's not transparent to us. We monitor changes, and they happen. Very small changes, but they are meaningful. And I think what we are creating for our single technology from Oxford is at least trackability. We can tell what has happened between each different software version. So maybe that's a different way, which is not currently popular.
Absolutely. So basically, once we build the thermometer, regardless of where you're going to stick it, let's just make sure the thermometer doesn't change. And that's what scanner upgrades do often.
Yes.
So that's why some transparency in this process will at least make sure that the thermometer itself is properly standardized. And I think the cardiac MR study group is, doing it the way that I was hoping for, which is here's an open source implementation, OpenMOLI, try it in different sites, and then try the commercial versions, see what happens. I think it's great follow-up work, and probably a good place to finish this. So thank you for all the discussion points. Thank you for joining. Thank you to those who contributed. I really enjoyed this discussion, and I hope I can be a listener on future MRM Journal Clubs. So thank you all, and have a good rest of the day.
Thank you.
Bye.